Good morning. My name is Amy Sullivan, and I'm the current immediate past president of AAAR. And it's my pleasure to welcome you to the 39th in our monthly AS&T lecture series. This is a new initiative for AAAR being supported by the Freelander Memorial Fund. So each month, the editors of our journal, AS&T, select a high-impact paper to be presented by its authors. With these lectures, we hope to be able to highlight the amazing research happening in our community, give an opportunity for all of us to come together outside of the annual conference, and also to tie our journal to other activities. In addition, each of these lectures is being hosted by one of our student chapters. With that, I want to thank all of you for joining us and everyone who's helped to make these happen. Thanks. And with that, I'll pass it over to our student chapter from the University of Miami to start us off. Thanks.
Hello, everyone. I'm Amin Shirkhani. I'm a PhD student at the Center for Aerosol Science and Technology from University of Miami. Today, it's my pleasure to introduce Dr. James Davis from the University of California, Riverside. Dr. Davis completed his Bachelor and Master of Science in Natural Sciences at the University of Cambridge, followed by a PhD in Chemistry from University of Bristol. He then carried out postdoctoral research at Lawrence Berkeley National Laboratory, and he is now the Associate Professor of Chemistry at UC Riverside. His research group focuses on understanding the physical and chemical characteristics of aerosol that influence their behavior and impact in the atmosphere using advanced techniques like spectroscopy, mass spectrometry, and interfacial chemistry. I would encourage everyone to ask their question in the chat during the talk so we can get to them after at the end of the lecture. So please join me in welcoming Dr. Davis for today's lecture.
Awesome. I mean, thank you for that introduction. And can everybody hear me? Just want to start by doing the classic Zoom thing. Yep, awesome. So thank you for the introduction, and thank you to Amy and the other folks involved in organizing these lectures for the invite to give this talk to you today. Getting a whole bunch of notifications on the screen. Just clear those. All right, so I am going to be telling you today about some of the work that we've been up to connecting the phase state of aerosol particles with their reactivity, aiming to understand how that phase state regulates the rates of reaction and the kinds of chemistry that can happen in organic aerosol in the atmosphere. I'd like to start by acknowledging all of the students that have contributed to the work that I'm going to present today. The paper, which is the focus of this talk, was lead authored by Ravleen Karakoli, who's a former graduate student in my lab. I'm also going to present some newer data from Stephanie and Craig, and this work was done broadly in collaboration with Ryan Davis, who's now at Sandia National Labs, and Kevin Wilson from the Lawrence Berkeley National Labs. For those of you who are unfamiliar with UC Riverside, I just wanted to share let me just get my laser pointer. Share a brief orientation. So Riverside is a city about 60 miles east of Los Angeles in the Southern California region. And it's a very beautiful location, pretty much surrounded by mountains. We've got several 10,000 foot plus mountains. We're not Colorado, but we do have some very beautiful natural landscapes nearby. On a very clear day, which is unfortunately not all that common in Southern California, you can see all the way to these mountains. And this next photo that I want to show is looking towards the San Gorgonio Mountains to the east. So this is very notable because it is somewhat of a rarity in Southern California. Just for a little bit of context, these hills in the foreground are about 6 miles away from this photo, and the mountains themselves are about 30 miles away. And you can see that they're both coming out in this photo very clean and crisp and clear. And this is owing to the fact that the air quality on this day was extremely good. There was very little particulate pollution in the atmosphere. For context, UC Riverside is just a little bit to the right of where this photo is pointing. Now, unfortunately, this kind of clear day is something of a rarity. We do have a lot of industry in Southern California, a lot of transportation, and this contributes to a lot of particulate pollution, a lot of VOCs, a lot of haze that can often impede both the air quality and the visibility. We also are very susceptible to fires. We're a very dry region. We're basically in a desert, and we are susceptible to significant fires, and oftentimes And these fires can be significant. So this fire was taken, this photo was taken from the Wikipedia page associated with this fire. This was the line fire, which burned back in the fall of 2024. This burned for about 40,000 acres in total. It was mostly contained after about 2 weeks, but it continued to burn for about 2 months before it was fully contained. And during this time, there were a lot of closures of schools, a lot of campuses switched to remote instruction because of the very significantly bad air quality as a result of all of the emissions from these fires. In fact, this fire was one of 3 which were burning at around the same time. The bridge fire to the north, the airport fire to the south, and the line fire to the northeast. Riverside was pretty much at the center point of a significant amount of fire activity at this time of the year. And so this can lead to some pretty bad environmental conditions. And looking at UCR's campus on a nice clear day, this turns into something which looks a little bit apocalyptic when these fires are burning and contributing a lot of absorbing particles and haze into the atmosphere. So as aerosol scientists, we're very interested in the structure and the composition of the particles that are emitted from these wildfires. And ideally, if we can take our magnifying glass and we can look at these particles, we can get a sense of what they're going to do in the atmosphere if we can characterize certain properties. For example, if we know the size distribution of the particles and the physical characteristics of those particles, if we know their composition and the chemistry that they can undergo, and if we know the environmental conditions, then we have pretty much everything that we need to know in order to understand how they're going to evolve in the atmosphere and the kinds of effects that they're going to have in the atmosphere. Now, taking a step back from just the air quality and kind of biomass burning motivation of aerosol research, we're all familiar with the role that aerosol play in the wider atmosphere in terms of their role in cloud formation, their interactions with light through both scattering and absorption. But aerosol are also useful in a lot of human-driven endeavors in industry and engineering, and in the synthesis of powders, for example, in the dispersion of chemicals in agriculture. And we also care about the role that aerosol have in health. So obviously the role that aerosol play in air quality is significant. We've also just lived through a pandemic where aerosol was strongly implicated in the spread of disease, but aerosol are also used to help treat diseases via direct delivery of drugs to the lungs. If we take our same magnifying glass to all of the aerosol in these different situations, these same characteristics come up as being the key things that we would need to know about our aerosol particles in order to predict both their effects in these situations as well as their evolution over time. As a chemist, I'm particularly interested in the chemistry of aerosol particles, and there are some very unique things that can happen in aerosol that allow some very unique kinds of chemistry to occur. And this is facilitated by the fact that aerosol particles, or an aerosol dispersion, has a very large surface to volume ratio as compared to the same amount of material dispersed in a bulk solution. So what this means is that you can get very rapid liquid vapor partitioning, and this can actually lead to some very interesting chemistry because it can drive chemical equilibria in an unfavorable direction. So as an example of that, if we take a weak acid, for example, just a carboxylic acid, in a particle that contains chloride, so this is something that you'd very routinely find in an ocean environment with sea spray aerosol, there'll be a very small equilibrium concentration of the carboxylate and HCl. So if you imagine kind of typical general chemistry, HCl is considered a strong acid. You would generally consider this equilibrium to be so far shifted to the left-hand side that you wouldn't even really treat it as an equilibrium. However, there are still small, tiny fractions of HCl that form in this equilibrium, and because HCl is volatile and we have this very rapid liquid vapor partitioning, this HCl can evaporate into the gas phase and disappear from the mixture. When we remove HCl from this equilibrium via Le Chatelier's principle, some more general chemistry popping up here, we actually drive this chemistry to the right-hand side, and this can actually have the effect of making this weak acid act like a strong acid. And this is a known effect. This is the reason that you get things like chloride depletion and ammonium depletion in particles. It's these weak acids or weak bases that, due to the liquid vapor partitioning, can act as the strong equivalents. This effect has also been observed to happen in respiratory aerosol. Bicarbonate is something which is present in respiratory particles, and this can actually transition to carbonic acid, which can break down to form carbon dioxide, which then immediately leaves the particles. And this can then lead to pH changes in these respiratory aerosol particles, and that's strongly connected to the evolution of the viability of viruses and bacteria within respiratory particles. Another facet of aerosol that allows some pretty unique chemistry or unique chemical environments is the fact that nucleation of solid phases is often impeded. And this can drive concentrations of ions in aqueous solutions to well above their solubility limits. A very familiar example, hopefully, to aerosol scientists is that of sodium chloride. So a sodium chloride solution in the bulk will saturate when you hit about 300 grams per liter. And this has a water activity of 0.75, or would be in equilibrium with a relative humidity of 75%. When you aerosolize that sodium chloride solution, you can retain that aqueous phase down to concentrations about 2 or 3 times, depending on your metric of concentration, about 2 or 3 times the solubility limit. So our sodium chloride droplet doesn't effloresce and form that solid state until you get down to about 40 to 45% relative humidity. So there's a huge region of relative humidity space where you have these ionic concentrations and these ionic strengths of the solution, which are far in excess of what you could ever measure in a bulk environment. Another consequence of this impeded nucleation, especially when you have organic material present in your aerosol particles, is that you can form very highly viscous phase states. So this is a figure from Koop et al. From 2011. I'm sure this has appeared in many, many talks over the years. This is a great demonstration of the range of viscosities that particles can exhibit in the atmosphere due to low humidities or low temperatures, ranging from the viscosity of just pure water at 10 to the minus 3 Pascal seconds, all the way up to things like 10 to the 8, 10 to the 10, where you're really getting into the regime where these particles appear to be solid, but they do have amorphous characteristics, and so they are technically still just viscous fluids. Now, the motivation for the work that I'm going to talk about today is really driven by this second point in how aerosol can facilitate unique chemistry. And what we really want to try to do is understand how the phase states that can form under these low humidity or low temperature conditions influence their reactivity towards oxidants in the atmosphere. So we're going to be talking about viscosity, we're going to be talking about phase transitions to states that impede reactivity in our aerosol particles. The strategy that we're going to adopt for conducting the experiments and to actually learn about this science is using single particle levitation. Single particle levitation is a pretty encompassing term for a suite of techniques that facilitate contactless levitation of aerosol particles. And the crucial point of single particle levitation is that contactless nature of the measurement. This means that we can preserve these states where nucleation is impeded. And so we can generate these very high concentrations of ions in our aqueous droplets, or we can form these highly viscous states because we don't have any contact with surfaces that can nucleate any solid phases. Single particle levitation can be achieved using a few different methods. Light is a very popular one. My PhD lab in Bristol with Jonathan Reed was the pioneering group in terms of developing optical tweezers for aerosol studies. You can use sound, acoustic levitation. This is typically used to levitate slightly bigger droplets. And you can use electric fields. And that is going to be the focus of my work and the focus of the slides going forwards. The advantage of these methods is that you can very rigorously control the environmental conditions. So you can control humidity, temperature, you can control the illumination, you can introduce oxidants, and so you can really rigorously mimic the conditions that aerosol particles experience in the atmosphere in order to understand how they're going to respond to those conditions. And finally, there have been a lot of analytical tools that have been developed, some by our lab, some by others, for actually characterizing the properties of these particles. And this involves things like elastic light scattering spectroscopy, Raman spectroscopy, microscopy, and mass spectrometry. The kind of electrostatic or electrodynamic levitation that we use is a descendant of the original Millikan oil drop experiment. So the Millikan oil drop experiment was one of the first demonstrations of electrostatic levitation. And this experiment was actually designed as a way to measure the charge of an electron. So Millikan wasn't really thinking about atmospheric chemistry or aerosol particles at the time. He was using and designing this experiment to measure something very fundamental of the universe, the charge of the electron. So there's been a lot of evolution of the methods over the years, and the kind of instrument that we use is now called the linear quadrupole electrodynamic balance. This is an electrodynamic balance which is based on this quadrupole configuration. We apply AC voltages in this this cross configuration, and this generates an electric field, which is what provides that lateral dynamic stability for our particles. And if this video works, this is kind of just a demonstration of the electric field oscillation inside the electrodynamic balance. So if you imagine sort of a macroscopic analogy, if you imagine a marble sitting on this saddle-shaped surface, under the influence of gravity, it can easily fall downhill, but it can't roll uphill. So it's stable in one direction, but it's unstable in the other direction. When you have the field oscillating, you can imagine that this field is essentially rotating. Every half cycle, the direction of stability is going to shift. And so whenever the particle or the marble decides to roll downhill, half a cycle later, it's going to get pulled back in to the center region of stability. And that's what holds it at the center of the trap. We also have other net forces acting. When you have this quadrupole configuration, this electric field provides lateral stability, but in order to provide axial stability or vertical stability, we do need to balance the net forces on the particle. And those net forces are the weight of the particle because it has a mass, and the drag force on the particle because we're introducing an airflow to control the environmental conditions. These electrodes are housed inside an aluminum chamber, and this helps us control the environmental conditions. We do have some limited temperature control. None of the measurements that I'm going to talk about today involve any temperature control. They're just under ambient temperatures. But we do control the environmental conditions through the relative humidity and through the oxidant environment that our samples are exposed to. To actually learn something about the particles, we need to use some analytical tools. So our analytical toolbox, once we've introduced our particles and we've controlled the environment by introducing the gas flow, we can illuminate them with LED light. So the LED is a broadband light source, and we introduce the light into the particle and we measure the light coming back off the particle. This yields a elastic scattering spectrum, and there are characteristic peaks in this spectrum which correspond to whispering gallery modes or morphology-dependent resonances, if you prefer, which are dependent on the size and the refractive index of our particle. And just to point out, the particle has to be spherical. So a lot of what I'm talking about when it comes to me resonance spectroscopy, we're dealing with aqueous or simply spherical liquid droplets. So this information gives us the size and the refractive index, and if we have things that are light absorbing, there are signatures in the spectrum that allow us to infer things about the light absorption characteristics of the particle as well. It doesn't tell us anything about the composition of our particle, and this is where we turn to mass spectrometry. So we've developed an open port sampling interface to couple with our linear quadrupole to allow our particles from the trap to be deposited into this open port sampling region and then sampled with a high-resolution mass spectrometer equipped with an electrospray ionization source. The way this open port sampling interface works is we have this T unit, which has these three kind of entrances or exits. We introduce our electrospray solvent from a syringe pump into the sidearm. That solvent comes into the T, it flows up to the top of the T, and if it wasn't for any other things going on, that solvent would just overflow and spill out into the lab. But what we have in this open port interface is an internal capillary which runs from the top of the T all the way out the bottom and to our electrospray ionization source. The ASI has this nebulizing gas, which helps you break up the droplets into ions for sampling into the mass spectrometer. But that nebulizing gas also creates a low pressure region at the tip of that ESI needle. So this actually sucks fluid along this capillary. So the functioning of this device is that the solvent comes in and it gets aspirated into the ESI via this Venturi effect, which is this effect of the nebulizing gases lowering the pressure and pulling the fluid into the ionization region. So what this allows us to do is to get a chemical snapshot of our particles as we release them one by one from the trap over any timeline we so choose. In this figure, we're looking at the oxidation of oleic acid, and we'll go through and we'll see more data that came from these experiments later. But each one of the peaks that you can see in this plot comes from a single particle that was deposited from the trap. We're tracking a mass peak corresponding to oleic acid and a mass peak corresponding to one of the products of the ozonolysis chemistry. And so you can see the oleic acid is decaying over time as it's being reacted away, and the product is growing in over time as more and more of that chemistry is happening. So this gives us the ability to determine the chemical composition and to look at the chemical evolution of our particles. We also do use bright field imaging. This allows us to get a nice picture of our droplets. We're not very good at this method, so we typically work with our collaborator Ryan Davis, who is an expert in doing coalescence measurements in droplets in this kind of electrodynamic balance. And this can tell us something about the viscosity of our particles. If you take two particles, you condition them under your experimental conditions, and you bring them together, they will interact and they will coalesce over a time scale which is going to be determined by the viscosity of those particles. So this gives us the ability to measure viscosity in our aerosol. I wanted to focus in a little bit more on the mass spectrometry side of things. This is often a part of the talk that I save to the end and then I run out of time. So I wanted to make it a little bit more front and center for this seminar. And we've been developing these mass spectrometry tools over several years. We initially developed them to look at the evaporation of multi-component aerosol particles. And so to that end, we were interested in characterizing particles that contained things with multiple different vapor pressures. So for example, in this example, we're looking at particles that contain triethylene glycol and tetraethylene glycol. We're characterizing their radius evolution over time as these two volatile components evaporate. And we're also tracking the chemical composition of the particle over time using our mass spectrometry method. So you can see that the tetraethylene glycol is evaporating more quickly because it has the higher vapor pressure. The tetraethylene glycol is evaporating a little bit more slowly. But crucially, what we can derive from this measurement is this compositionally resolved evaporation, which is facilitated by having this mass spectrometry technique. And we can verify and validate all of this through an evaporation model. So the vapor pressures can be fed into it, a simple gas phase diffusion limited evaporation model. And that's shown by these dashed lines. So we can predict both the radius evolution due to the evaporation, as well as the specific chemical evolution of the particle. And you can see that we get agreement across all of the measurements that we made on this system. It was a little bit after this that we turned our attention to reactive systems, looking at some heterogeneous chemistry. And we focused on ozonolysis chemistry because it's fairly well established that one can make controlled amounts of ozone in a single particle levitation device. And so what our mass spectrometry tools allow us to do is to characterize the kinetics of that ozonolysis chemistry, as well as get a snapshot of the chemical composition of our particles both before and after that chemistry is occurring. So again, looking at this oleic acid ozonolysis example, we can get a chemical snapshot of how the system is changing over time under different conditions that we can control. So as an overview for the rest of the talk, we're going to be focusing on two areas that have kind of the same themes associated with them. The first we're going to talk about is the main focus of this AS&T article, which is meant to be the highlight of this seminar. And this was work that we did in collaboration with Dr. Kevin Wilson at the Lawrence Berkeley National Labs, and it was spearheaded by my graduate student, Ravleen, who was graduating in this photo of her. So to start with, just a very brief overview of ozonolysis chemistry. So when you have organic molecules that have a double bond, they are susceptible to reaction with ozone. The ozone adds to the double bond to form this malozonide, which can then decompose to form your Kriege intermediate and a carbonyl. And so this chemistry is pretty well established. And for the systems that we're going to be exploring, the products are very well known and very well characterized. The rate of this ozonolysis chemistry depends on a few factors that are dependent both on the kind of inherent molecular reactivity of our organic molecule, and that's going to depend on the functional groups and the steric hindrance of that ozone accessing the double bond. But the reactivity is also going to depend on the particle properties. So for example, the size of the particles as well as the characteristics of the fluid itself, so the viscosity and how that affects the diffusion of both ozone and reactants in the particles. So we were interested in exploring how different phase states affect this kind of chemistry, and we wanted to start by looking at a very straightforward comparison between oxidation of a liquid versus oxidation of a solid. But we wanted to choose a sample system where we didn't have to worry too much about the inherent reactivity of the organic molecules. And so we turned to oleic acid because oleic acid is a liquid at room temperature, and this is the cis isomer of the molecule. The trans isomer is elaidic acid, and that's actually a solid at room temperature. And there have been studies in the past that show that the reactivity of the double bond in these two isomers is very similar. So this gives us the opportunity to study a molecule, two molecules which are very similar to one another, but that exist in two different phase states. So starting with our look at oleic acid, you've seen some of this data already. We can vary the ozone concentration and we see the different kinetics, and we can establish things like uptake coefficients, and everything is kind of consistent with what you expect from the huge amount of literature that has gone into studying oleic acid over the years. We did see some interesting effects when it came to our mu resonance spectroscopy method. So this plot shows the radius evolution in the black points and the refractive index evolution in the red crosses. And crucially, about 500 seconds into the oxidation in this particular experiment, and this I think was from the red curve at 2 ppm of ozone. But crucially, after a few hundreds of seconds of oxidation, we get this huge upswing of the refractive index and a pretty significant breakdown of the ability to derive the size and the refractive index. And this is shown by the change in the spectra in the plot in this lower portion. The black or the darker line shows the spectrum before, where you can resolve these very sharp peaks. But after oxidation occurred, you get this broadening of these peaks, and it just generally looks a lot more noisy. And we attribute this to liquid-liquid phase separation. So it's not full-on solidification. What we think is happening, based on other literature that's explored this kind of effect, is that the products of the ozonolysis chemistry are actually phase separating out from the oleic acid liquid itself and possibly forming islands on the surface of the particle, which are large enough to disrupt that spherical symmetry that we need to support these morphology-dependent resonances, but not enough to affect the kinetics. We don't see any effect on the kinetics due to this apparent phase transition that is occurring. So this is consistent with forming these liquid islands that don't really impede uptake of ozone, but do break down our mu resonance spectra. So some interesting effects going on with oleic acid, a lot of it just consistent with previous observations of the ozonolysis chemistry of this system. So now turning to elaidic acid is a much lesser studied organic molecule. It is a solid at room temperature, so in our minds, we are going to be able to make particles with this solid form and then compare the oxidation chemistry to the liquid form, which was found in our oleic acid. What we found, though, is that the reaction kinetics looked pretty much identical. This plot shows, I don't remember the exact ozone concentration in these two experiments, but it was the same between them. This shows the decay of the oleic acid on the left and the decay of elaidic acid on the right, and the formation of some of the products. And you can see that, at least from the decay of the acid, we're getting very, very similar reaction kinetics. There are some differences in the products. I'm not really going to go into the products in too much detail, mainly because we don't have a good handle on the quantitative aspects of how much product we're forming. But what is really interesting here is that this suggests that either the solid form of our elaidic acid is reacting at the same rate as our liquid, or we're forming a liquid elaidic particle at room temperature. And when we turn to our Mi-resonance spectra, it is indeed the case that our elaidic acid is remaining a liquid, even though we're significantly below its melting temperature. It should be a solid thermodynamically under the conditions of these experiments. What you will notice about this plot on the right, where we're looking at the radius and the refractive index evolution once again, is that we're getting a much more kind of even trend. There's no breakdown, there's no discontinuity and big change in the refractive index. And this suggests that then the products of the elaidic acid oxidation chemistry are actually well mixed within the particle. So we're not getting this phase separation effect that we saw with our oleic acid. So all evidence points to our elaidic acid remaining in this liquid phase state, which is in essence a supercooled state, because we are now we're now liquid in the temperature range where we would expect our solid phase. So this is a pretty nice example of how the aerosol particles impede the nucleation of a solid phase, and this isn't just going to happen in aqueous salts; it happens in these organic liquid systems as well. So it was possible to observe solidification in our elaidic acid particles. It wasn't something that we were able to control. It seemed to be a pretty stochastic effect. But when we were able to generate solid particles in our linear quadrupole trap, we were able to identify significant changes in the oxidation kinetics. And so this plot on the left shows our single particle experiments. The red points show our supercooled liquid particle, and the black points show particles that were actually solidified inside our chamber. And you can see that there is a significant slowing of the reaction consistent with the formation of this solid phase state. This is when we reached out to Kevin Wilson at the Lawrence Berkeley National Labs, and it turned out that they were also studying elaidic acid. We hadn't talked beforehand about this, but they had some flow tube measurements of elaidic acid where they were controlling the temperature of their reaction. And what they found was that at 20 degrees, so at room temperature, they saw very rapid oxidation chemistry, but then at minus 20 degrees Celsius, they saw very slow oxidation chemistry. So there seemed to be a phase state change going on between these two temperatures, which is consistent with a freezing transition occurring. So our higher temperature elaidic acid is liquid, and our low temperature elaidic acid is basically being frozen out. We've lowered the temperature enough that driving force for nucleation of the solid phase allowed it to actually solidify. And so we can identify in this case a significant slowing of the reaction chemistry. We did use a multi-layer chemical kinetics model, the KM gap model, to do some kind of pretty crude simulations to get an understanding as to whether this was indeed explainable through diffusion limitations, and broadly we can reproduce the kinetics here with lower diffusion coefficients for the ozone and the reactants in our particles when we have these solid states. So the kind of conclusion of this work is that microdroplets, aerosol particles, can allow fluid states to exist even when you would thermodynamically expect a solid state to exist. And so this is something that is always going to be something that we have to think about. We have to know the history of our particles in order to be able to predict what kind of phase state they might be exhibiting. The second half of this talk is going to, or the second half of the results of the talk, is going to concern more relative humidity dependent phase state changes. So we're still interested in phase states. We're still interested in how those phase states affect chemistry. But in this part of the talk, we're going to be focusing on much more oxidized organic material and the role that salts play in controlling the phase states of our systems. This was work done in collaboration with Ryan Davis. His students at Trinity University, when he was still at Trinity, were heavily involved in this work, as well as my students, Stephanie and Craig, from the UCR end of this collaboration. So taking a little bit of a step back, we spent some time a few years ago trying to understand what happens when you add inorganic co-solutes to organic, aqueous organic particles and how they can regulate the phase state or the characteristics of the particles. So we were looking at a particle mixture that contains citric acid and ammonium sulfate. And we have methods in our lab using resonance spectroscopy to estimate the diffusion coefficient of water. I can talk about that a little bit more if you have questions. But what we found in this system of citric acid and ammonium sulfate is that the diffusion coefficient of water decreases strongly with the relative humidity because it gets more viscous. But we didn't see a strong compositional dependence. So when we added in ammonium sulfate, we didn't really perturb the water diffusion coefficient in any appreciable manner. We also made measurements of viscosity. Well, Ryan Davis made the measurements of viscosity. And what they found was pretty starkly different than what our diffusion measurements indicate. So what they found is that in general, as you go to lower humidity, the viscosity increases. But when you add in ammonium sulfate, you get these huge order of magnitude steps up in the viscosity with even fairly small amounts of ammonium sulfate added into these particles. So we were interested in exploring what's actually going on in this connection between diffusion and viscosity. Now, typically, viscosity and diffusion coefficients can be connected through the Stokes-Einstein relationship. This is a pretty crude relationship. It makes some assumptions about the size of the molecules and the things that those molecules are diffusing in, but it is pretty common to connect diffusion and viscosity using this relationship. When we plot our diffusion coefficient and our viscosity together for the different samples that we looked at, we get these colored lines, the black, the red, the blue, and the green. And if we were agreeing or if we were following the Stokes-Einstein relationship, we would actually be falling on this line all the way at the lower left of this plot. These contours on the plot are the contours of our fractional Stokes-Einstein relationship. So it's pretty well understood that the Stokes-Einstein relationship breaks down for these kinds of mixtures. And so the fractional Stokes-Einstein relationship introduces this exponent in order to essentially try to account for some of the assumptions and the limitations inherent in that Stokes-Einstein relationship. So when you reduce the value of this exponent from 1, which would be pure Stokes-Einstein, to 0.8, 0.6, 0.4, you essentially build up these contours in this diffusion versus viscosity space. And so you can see that our data is more consistent with this fractional Stokes-Einstein relationship when you have an exponent between about 0.7 and 0.5. And Alan Bertram's group has done a lot of work trying to connect the value of this exponent to some molecular characteristics of our system. And so they empirically connected the value of that exponent to the relative size of your diffusing molecule to the size of the molecules in the matrix in which they're diffusing. So for in the case of water diffusion in citric acid, our diffusion would be the size of a water molecule, and our matrix would be the size of a citric acid molecule. And indeed, when we're dealing with pure citric acid and we use this empirical relationship, we find that our data is in agreement with this R matrix value being the size of just a citric acid molecule. When we add in our ammonium sulfate, we find that this R matrix value is getting bigger, much bigger than the sulfate or the ammonium that we're adding in. And so what we think is happening here is that there is some aggregation or some interaction between our salt ions with the organic that essentially causes this aggregation, and it causes the viscous matrix to appear to be made up of bigger regions. And so in essence, this is why our water diffusion coefficient doesn't really change, because it can still percolate around these larger clusters that are forming inside our fluid. Okay, so we do see this effect with ammonium sulfate and citric acid. We don't really see a phase transition here, but we do see a continuous perturbation to the physical characteristics. If we replace our ammonium sulfate with something which contains a divalent cation, so magnesium or calcium, we can actually induce a phase transition in these organic-inorganic mixtures. And these measurements were again performed by Ryan Davis through coalescence. And he was looking at a series of these sugar alcohol molecules, and they are just very oxygenated organic molecules. And what he found is that the pure sugar alcohols, as a function of relative humidity, became moderately viscous. But as soon as he added in calcium chloride, the viscosity characteristics changed drastically. There was a significant increase in the viscosity, even at relatively high relative humidities. And crucially, in all of these cases, there was a step transition in the viscosity to something which resembled a rigid particle. So the points that you can see at the very top of these plots essentially correspond to coalescence events that didn't ever coalesce. So it was taking two rigid particles, bringing them together, and then nothing was happening. So what's happening in these cases is a phase transition to something that we call a gel. And this is a solid liquid phase state. So the gel formation process is associated with the formation of this rigid structure that has pores and channels in it. So it entrains a fluid phase within that rigid network. And we can verify this using Raman to see that there's still water present. We can do water diffusion measurements to show that there's still water transporting back and forth. But it is a new phase state that forms. So we wanted to understand what happens when you form this gel when it comes to reactivity. So none of the species that Ryan had looked at in that earlier work would be reactive towards ozone. And so we turn towards ascorbic acid. This is vitamin C for any of you who take supplements. Vitamin C is an antioxidant. It has a double bond, so it is susceptible to reaction with ozone. And we were able to characterize its viscosity as a function of humidity, its water diffusion characteristics, its hygroscopic growth, and everything points towards our ascorbic acid forming a gel when it is present with calcium chloride, and on its own it forms a viscous fluid. So what this allows us to do is to compare the oxidation of ascorbic acid when it's present in a gel state versus when it's present in a viscous phase state. And so we can start to hopefully see differences between our gel state and our viscous phase states. So just briefly covering the evidence that we have for gel formation, this panel A, we have this step change in viscosity, same as what we saw in the previous slide when we have our calcium chloride present. And crucially, when we have ammonium sulfate or just pure ascorbic acid, we don't really see too much effect on that viscosity. And we don't even see the effect that we saw with the citric acid. From the diffusivity point of view, the diffusion coefficient of water, we see this general decrease with relative humidity, but a step decrease with the onset of gel formation. This panel C shows something which is essentially a measure of the sensitivity to a change in the relative humidity. It is how big the particle's response is to a step change of 1%. And so you can see again for the pure ascorbic acid and the ascorbic acid with ammonium sulfate, a fairly continuous change in this parameter. But for our calcium chloride mixture, we see a step transition. And again, this is occurring where we expect our gel formation point to be. And finally, this Stokes-Einstein plot, you can see that there's a significant deviation of the behavior of our gel compared to the behavior of our viscous particles. So the addition of our ammonium sulfate, when we're focusing on this Stokes-Einstein plot, does lead to a decrease in that exponent, which is consistent with what we saw with the citric acid and the ammonium sulfate. But crucially, we don't see any change of phase state. We only see this gel formation or this phase transition when we have calcium present. So we wanted to see what happens when these particles are exposed to ozone. So this comparison between viscous phase states and gel phase states hasn't been explored previously. And I want to point out that this is work that's currently under review. So if you are one of the reviewers, please be nice. At high relative humidity, we don't really see a huge amount of difference between the oxidation of the ascorbic acid in these different particle mixtures. There are some different kinetics here that we can reasonably attribute to the amount of water that's present in the particles. However, when we go to low humidity, we start to see significant deviations in their behavior. The reactivity of our ascorbic acid with ammonium sulfate is pretty fast, and this is consistent with our water diffusion data that suggests that even under these lower humidity conditions, there's still pretty rapid water diffusion. Our pure ascorbic acid, which is forming this viscous fluid, is much more slowly reacting, but there is a continuous reaction over the whole course of the experiment. Our gel particle in the green shows pretty unique reactivity in that you get this rapid reaction initially, followed by a straight up plateau. So there's no further reaction happening after that initial region when we have our gel state present. So focusing on that gel forming system a little bit more closely, looking at the different relative humidities for our ascorbic acid with calcium chloride, you can see that there is a slowing as we decrease the relative humidity, but it's that transition from 30% to 25% that we get this abrupt change in the reaction kinetics and the kinds of kinetics that we're seeing. And this is where that gel formation is happening. So our gel particles are showing this kind of plateauing effect, and we attribute this to essentially the phase state or the structure that's forming in these gel particles. So the gel is this solid liquid mixture. The solid is this porous structure, this porous rigid skeleton filled with a fluid. And so what we think is going on here is that the ozone is able to penetrate into the fluid-rich pores and react with the ascorbic acid in the fluid phase, but it can't react with the ascorbic acid in the solid phase. And so that's why you get this plateauing effect, because the rest of the ascorbic acid is protected and locked up in that solid particle. So that's broadly kind of shown here. This comparison between our viscous state, where you have this slow transport through what is a pretty homogeneous system, leads to a reaction that is slow, but does continue consistently with time. And this is what was going on with our pure ascorbic acid, but our ascorbic acid with our calcium chloride, we're forming this gel state. And this gel state has this kind of rigid network of ascorbic acid bridged by our divalent cation, the calcium, and then the pores are essentially where that fluid phase exists. And so in the picture that we've developed, you have ozone able to penetrate into these pores and react with ascorbic acid, but it can't react with the ascorbic acid that's kind of bundled up in that solid network. So with that, I'd like to just provide some takeaways from both points of this talk, and then I'll shut up and answer some questions. In the first part of the talk, I showed that the phase state of the particle isn't something that we can always easily predict, and the solidification or nucleation of a solid phase is a stochastic process that is not always going to happen in aerosol particles until you have a very large thermodynamic driving force. We showed that these liquid organic particles freeze at much lower temperatures than in the bulk, and this is expected. This is something that's seen in water, for example. You don't get homogeneous freezing of liquid water until you get to around -30 Celsius. But we were able to identify that these different phase states had profound effects on the reactivity, and this was linked to the diffusivity of material within the two different phase states. Moving to the conclusions of the second part of this talk, we demonstrated that when you have inorganic species present in mixtures with your organic materials, the interactions between these species can play a pretty definitive role in regulating the phase state. You can form these gel states, which are an example of this liquid-solid phase separation effect, driven by the supramolecular network of divalent cations and oxygenated organic molecules. And we showed that the gel state has a pretty significantly different heterogeneous reactivity than the viscous phase state for the same material. And this is pretty important because we think that gel states are probably a lot more prevalent in the atmosphere than is currently known, because you do find these divalent cations in sea spray, you do find a lot of oxygenated organic material in aerosol particles, and so we expect these gel states to be pretty common, if not necessarily as abundant as just simply viscous phase states. But crucially, experimentally, it's quite difficult to tell the difference between a gel state and a viscous state when you're doing ambient sampling. Bounce factors, for example, will more or less show the same between a gel state and a viscous state. So hopefully this is going to prompt more work to try to identify the differences between these methods from an ambient sampling perspective. So with that, I'd like to acknowledge the funders of the work in my lab. I think everything I've talked about today has been funded by the NSF, with some of the early work funded by a fellowship from the American Society for Mass Spectrometry, the ASMS Foundation. I'd like to thank my collaborators, Ryan and Kevin, and their students and postdocs that were involved in this work. And I will happily take any questions from the audience.
Thank you so much, Dr. Davis. So the floor is open right now. So please simply unmute yourself and ask the questions. And I'll also check the question in the chat. So there is one question in the chat. Do you expect a gel state for a multi-component mixture in salts and organics?
I think yes. This isn't something that we've looked at. So obviously these are multi-component mixtures, and that you have kind of a salt and a single organic. Ambient particles are going to contain a lot of different organics and potentially a lot of different salts. We haven't studied how the presence of other salts might impede the formation of the gel. If you imagine sea spray is predominantly sodium chloride, but there is some calcium, there is some magnesium. Does that large amount of sodium chloride override this gel formation effect? Some of Ryan's earlier work did show that you really only need small amounts of calcium or magnesium to form these gels and see these big changes. But as to what other species present might do in kind of interfering with that process, that's still an open question. Thanks for the question, Uli.
Any other question?
James, this is Vasanthi from Naval Research Lab. I have a question on one of your slides where you're discussing the index of refraction measurements. Are you making these measurements at a single wavelength? I guess I'm trying to figure out how you're decoupling the size versus refractive index measurement using a single wavelength.
So we're not using a single wavelength. So the scattering spectroscopy that we do uses an LED that has a broadband output. So a lot of the measurements that we do use a red LED, which is emitting between about 640 and 680 nanometers. So we're collecting the scattering spectrum over a pretty, well, not a wide angular range, but a wide enough angular range to resolve a lot of features in the scattering spectrum. And then we can essentially fit the positions of those using me theory and some of the algorithms that Thomas Preston developed. And essentially that's able to deconvolute the effects of both the size and the refractive index and yield information on both of them from a single spectrum.
Yeah, okay, all right. Yeah, thanks.
So there's another question in the chat. Missed the part about the fractional Stokes-Einstein equation. May I ask if you think the equation is not valid when it becomes highly viscous? So I think the fractional Stokes-Einstein equation does work at high viscosities. I think there are deviations. Let me just bring back the slide that showed some of that. Yeah, so this fractional Stokes-Einstein equation, for something which is a single component, for example, water and citric acid, or water and sucrose, you can use this fractional Stokes-Einstein equation across pretty much the full viscosity range with a single exponent value. And I think this is because as you get more and more viscous, you're still forming that viscous fluid from molecules which are the same size. And so it's only when you have additional components present that might interact with your viscous forming molecules that you start to see not so much a breakdown, but a composition and a compositionally developed, a compositionally dependent exponent value. In that, as you go to lower humidities, for example, you might get stronger interactions between the ions and the organic molecules present that then force this parameter to deviate more as a function of the composition. So I think the fractional Stokes-Einstein equation, it is valid in the kind of in the sort of empirical space that it's been developed. But it does require kind of some additional dependencies to fully capture the behaviors. And then it becomes less and less clear if you're just fitting your data with a bunch of parameters. And then the question comes is, do those parameters have any physical meaning anymore? So I don't know if that fully answers your question, but the sort of TLDR, yes, I think it does apply still at high viscosities.
Hi, I'm sorry, can you hear me? Maybe
Yeah.
I just like specific my question because I also like use the fractional Stokes-Einstein relation to do simulation. Like we have like viscosity prediction, which should be very good. I'm in Andy's group, like just AOMFAC Visc. I'm not sure if you heard of this model. We just use it to predict the viscosity. And when we just starting, when like the viscosity reached to like higher than 10 to power 8, and just like the viscosity, the diffusivity, what we predicted is much more lower than the measurements. And I don't understand why. Um, yeah, just because there's no measurements for that. So I'm not sure if you have any measurements like when you have like the viscosity of those high viscous flow, and you also have corresponding diffusivity, which predict by like the fractional Stokes-Einstein, and you also have the measurements of the diffusivity. Do you have any experiments about that?
So we have the data for the citric acid, which is what I presented. So there is a pretty well established breakdown of the Stokes-Einstein relationship when it comes to a small molecule diffusing in a viscous matrix made up of larger molecules. So the Stokes-Einstein relationship is an approximation. It's a model, and it works best when you have a big diffusing sphere through a continuum phase. When you're looking at something like the diffusivity of water, water is very small compared to the size of the molecules that it's diffusing through. And so it's pretty well established from measurements that if you just apply the Stokes-Einstein relationship using your measured viscosity, your water diffusion coefficient will be orders of magnitude faster. And the reason for that is that water is a small molecule that can kind of scoot around the molecules in your viscous matrix. The fractional Stokes-Einstein does kind of correct for that because it considers the size difference. But this is an empirical correction, so it's not derived from theory. It is an empirical correction that works pretty well, but may lose some predictive power in certain situations. As to your comment on the AIMFAC Visc model, I've never used the AIMFAC Visc model. I am very familiar with AIMFAC and all of the work that goes into that, but that's not something that we used to kind of compare to any of the measurements that we had here.
Okay, I see. Like because I was missing this part. I was wait. I'm sorry. I just find your figure like the highest viscosity you reach is 10 to power of 6, right? For this figure, so it's not super viscous.
It's not super viscous, no. I think the
Okay.
viscosities reached up to about 10 to the 8 for
Yeah.
some
Yeah.
class samples.
Yes.
But
I
they kind of
just
spanned out a pretty broad range.
yes. I see. Just based on my research, when the viscosity reached to higher than 10 to power of 8, just like I feel like I don't know, just the fractional Stokes-Einstein is no longer valid.
Okay,
Yeah, just I just want to ask. Thank you very much. It's a nice talk.
thank you. So there's another question in the chat. I'm wondering why divalent salts form a gel while monovalent salts don't. I think this comes from the coordination of the organic molecule kind of around the salt. The higher charge leads to kind of stronger interactions, and we think it can facilitate kind of more bonding points. And so the formation of hydrogels with calcium is pretty well studied, and it kind of just allows this three-dimensional bridging structure to form. Whereas a monovalent salt, I don't think you can necessarily coordinate as many organic molecules as strongly to form this kind of large supramolecular structure. Hopefully that answers your question, at least to the limit of my knowledge on the molecular details of this process.
All right, so if there is no question, let's thank our speaker again. And thank you all for attending today's lecture series. And meet you all in the next lecture.
Thank you very much, everybody. Have a great day.